Thank you very much. It's really a pleasure to be here. I want to talk about what I think is, let's see if this works, I think is really one of the major problems we face, which is, there we go, which is how to connect the architecture of regulatory circuits to the behavior of cells. What is it about this pattern of interactions, who regulates who and how, that allows us to understand, or could allow us to understand, how cells actually behave? And in my lab, we focus on the question of dynamics, which I think is one of the real things that connects these two levels of thinking about cells. So the idea here is that in order to understand how the architecture of a circuit really affects what the cell does, we really have to study the dynamic behavior of proteins and genes at the level of individual cells. And the examples I'm going to give you today all kind of are focused around that point. Okay. So the way we do it in the lab is we really have three techniques. We use time-lapse movies to follow the dynamics in individual cells. We use synthetic biology to construct or rewire genetic circuits so that we can test particular hypotheses about different architectures. And we use mathematical modeling to connect the level of the genetic circuit architecture to the predicted dynamics. Okay. Now, what I want to start with today is a very simple observation in many systems that signals regulate the cell by activating transcription factors. And so you often have a case where a signal will activate a transcription factor, which will in turn activate target genes, and this will lead to some kind of dose-response curve. Okay. Now, the interesting thing that's emerged recently is that for many of these systems, even when the input is held perfectly constant, the output is not constant. The output comes in a series of what look like stochastic coherent pulses, where many or all of the molecules in the cell will suddenly be activated for a short time and then deactivated again. And these pulses go on. We see this in NF-kappa B. We see this in P53 through some recent beautiful work from Galit Lahav's lab in particular. We discovered this a few years ago in yeast with the calcium response pathway mediated by the crazy one transcription factor, seen in a variety of different systems. Okay. And it raises several, And it raises, I think, several basic questions. First, how general is this behavior? Do we see this kind of pulsing in all kinds of cells or only in eukaryotic cells, for example? Second, how do cells do this? How do they take a constant input signal and turn it into a pulsatile output signal? And I think most important for me, anyway, is the question of why. What is good about pulsatile signaling in cells? What can you do with this mode of regulation that you couldn't do with otherwise? Okay. To address these questions, I want to talk about three projects that are all in the same organism. This is Bacillus subtilis. It's a bacteria. It's kind of the #2 best studied bacteria out there. And we love it because if you put them in constant conditions, what you see is that these cells act very individualistically. They activate a whole range of different responses to stress. And I'm going to talk a lot about responses to stress today. And so what you can see in this movie is individual cells in the same condition, some growing and dividing in green, some turning into competent cells. Those are the red labeled cells in the middle that you see transiently going into that state, and some turning into spores. And I'll talk more about sporulation in a few minutes. Okay. So having seen that kind of dynamics in a number of experiments, we wondered what other dynamics might be hidden in this organism. And James Locke and John Young decided to look at a variety of other systems in Bacillus. And the first thing they looked at was the general stress response. So the general stress response in bacteria is mediated by an alternative sigma factor called sigma B. So alternative sigma factors are specialized transcription factors that recruit the polymerase and bring it to a whole set of target promoters. Okay? And there's a lot of these. They're highly conserved. They're very central players. Now, in sigma B, stresses post-translationally activate sigma B, and that in turn leads to the activation of a whole range of different targets. Okay. So the first thing we did was we just took cells and we put in a yellow fluorescent protein reporter for sigma B activity, and we made movies under conditions where the cells have an energy stress that reduces internal ATP levels. So when you do that, what you see very strikingly is stochastic pulses of activity of sigma B. And if we plot the rate of production of YFP, which is a measure of the promoter activity at any given time from cells in these movies using tracking algorithms to follow individual cells, we see something very striking, which is the promoter activity of a sigma B target. It comes on in pulses that are very stochastic. So these are three different lineages, for example. You can see this kind of pulsy dynamics. Okay. Now, if you ask, how does the magnitude of the stress affect these pulse dynamics? It's very interesting because it doesn't really affect the amplitude or duration of the pulses, but it has a very strong effect on the frequency of the pulses. So we can say this system is a frequency-modulated stochastic pulsing system. And the first question we want to ask is how does this work? How does the cell turn a constant input level into a frequency of stochastic pulses of activation? And to think about that, we were struck by the parent stochasticity, the erraticness of these pulses. And we've always been interested in the question of noise. So as we know now, in cells, a lot of behaviors and a lot of processes like gene expression can be inherently stochastic. That's very easy to see. This is an image from a paper we did about 10 years ago now, where you just take 2 different copies of green fluorescent protein that are 2 different colors. You put them under the control of identical regulatory sequences, same promoters, in the same cell, and then you ask whether the cell expresses them to the same level. And it's very easy to see that stochasticity in the expression of genes leads to a lot of variability in protein levels in cells. And the reason we care about that is because the question is, to what extent or how does this impact the behavior of this kind of stochastic pulsing system? And you can imagine 2 distinct models. In one case, you might imagine that what the cell is trying to do is have a very regular oscillator, and the noisiness of the cell is really degrading that behavior and making it look very erratic. The alternative is that noise is really necessary for this kind of pulsing. That noise is an integral part of the pulsing mechanism, and that without noise, you would not have pulses at all. Now, whenever you think about noise, it's really a slippery subject, because the problem is, how do you tell if something is due to noise or if something is due to some other component in the cell that you just didn't look at? And one way to get at this is with a simple idea, which is if you want to get rid of noise in cells or reduce it, why not just make cells a little bit longer? So if they get longer, if you do it in the right way, you can make a mutation that prevents septation of cells. The cell size gets larger, but the density of nucleoids and the concentrations of proteins don't really change. And so the fluctuations go down without affecting the mean behaviors of most cellular processes. So if you do that, you can make movies like this and ask whether you still see pulsing in very long cells. And the answer is that pulsing is suppressed. And so this suggests the idea that noise is really required for this pulse generation mechanism. So to better understand the mechanism, you actually have to think about the components that are involved. And sigma B has a very well-studied regulatory system that involves an anti-sigma factor, we'll call W, that binds to and inactivates sigma B, and an anti-anti-sigma factor called V. And the idea here is that V is a phosphoprotein, and the anti-sigma factor W can phosphorylate V. When W interacts with V, that suppresses W's ability to inactivate sigma B, as I've drawn here. Okay. Now, how does this system get activated by stress? Well, there's a phosphatase that comes in and dephosphorylates the anti-anti-sigma factor. That leads to this inactivation of the anti-sigma factor and release of sigma B, which can go and activate the system. There's one other interesting aspect of this circuit, which is that there's a feedback loop here where sigma B activates its own operon and therefore activates its own gene as well as the genes for the anti and the anti-anti sigma factors. So this is a mixed feedback loop that has both positive and negative effects on the system. So what's going on in this circuit? How does this thing work? Well, through a bunch of experiments, which I'm only going to show you a couple of examples here, we realized that this first module is a phosphoswitch. So you have 2 enzymes, a kinase and a phosphatase, working against each other. And many years ago, I think almost 30 years ago now, Goldbeater and Koshland suggested that this kind of motif could function as an ultrasensitive switch. And so we engineered a version of this circuit where we could analyze the system open loop, and we could see that indeed this module operates like a very ultrasensitive switch. Okay? Now, the reason that's interesting is because if there's fluctuations or noise that affect the ratio of the kinase to phosphatase, that can give you a big change in the activity of sigma B. The second part of this circuit is the feedback module. And the role that this can play is by having both positive and negative feedbacks, you can first amplify a pulse once it starts and then terminate it. Okay, so this is how this might work. Imagine for a second that the system is off, and then there's a fluctuation where the phosphatase levels suddenly go up. That leads to a big pulse in sigma B activity, followed by production of the anti-sigma factor in red here, which shuts the system back off again. So this is a pulse generation and termination mechanism, and we wanted to test it inside cells. And I just want to give you a flavor for the kinds of experiments we do to test these ideas. So here we made an open loop version of this circuit where we knocked out the feedback loop, so we prevent sigma B from activating its own operon. And now you can play this game. you can set the phosphatase levels to a constant induced level, and then play with and suddenly turn on the operon, including the anti-sigma factor. You can turn it on to different levels. If you turn it on to levels that don't cross the threshold set by the phosphatase, then sigma B turns on and stays on. But if you go up even higher to where it crosses, you see that you get this nice pulse shape where as long as the phosphatase level exceeds the kinase level, you're on, and then once they cross, it turns off again. This makes predictions that you can test directly experimentally. As we vary the level of expression of the operon, we expect to see at first the system should turn on and stay on, and then later at stronger values of operon expression, it should turn on, pulse, and turn off again. And indeed, you get very similar behavior in the simulation and the experimental results. Okay? So that suggests that this really is the pulse generation and termination mechanism. That leaves one more question for the mechanism, which is, now how do you control the frequency of these pulses? And we were struck in the experimental data that when we changed the energy stress that the cell was feeling, it changed the mean expression level of the phosphatase itself, suggesting that what was really controlling the frequency of pulses was just the phosphatase expression. And so that would look like, this mechanism would look like this. At low values of stress, the phosphatase would be low, and you would occasionally get a threshold crossing event, which would trigger a pulse. At higher values of mean phosphatase expression, you'd get more of these threshold crossing events just due to noise, and you'd get a higher frequency of pulses. Now, to test this idea, we really wanted to get rid of the normal energy stress sensor itself, because we want to see whether this frequency modulation mechanism can work independent of any actual inputs we might not be aware of that go into the system. And so we took advantage of the fact that I'm going to tell you more about in a second, that there's a second input into this pathway from what are called environmental stresses, things like salt and alcohol and things like that. And it goes through a big 1.8 megadalton complex called the stressosome that triggers activation of another phosphatase, which we'll call TU. So now we can do this. We can get rid of the normal energy stress sensor. We can get rid of the stressosome. That makes TU constitutively active. We can get rid of TU too. And then we can put in our own TU under the control of an inducible promoter. And in this way, we can ask whether, as we vary the expression level of a phosphatase, that's sufficient to give us frequency modulation. And so we made movies of this heavily rewired version of the system. We see very similar dynamic behavior with pulsing, and we see exactly the kind of frequency modulation we get in the endogenous system. Okay. So what does this tell us? This tells us that if you want to make a DC to AC converter, that is, something that takes a steady input level and converts it to a frequency of stochastic pulses, it doesn't take very much. You can do it with this simple 3-gene system that has these interesting post-translational interactions, this phosphoswitch and the feedbacks, and so on. So this is a motif for that. But we were very happy with that, because we thought, OK, this is how you do it. Now, at that point, John pointed out that we really only understood half of the system. Why? Because I just mentioned there's that whole stressosome input. What is that thing doing there? That's not necessary for this kind of pulse behavior. So if you think about the full pathway, you can start making movies where you knock out the energy stress inputs and instead focus on the environmental inputs. So this is a movie where you give the cells a big step of ethanol. So you just go from 0 to 2%, and you watch what happens. And I think what you can see in this movie is that it's complete opposite of what we get with the energy stresses. So here, instead of stochastic, erratic, unsynchronized behavior between individual cells, we get all the cells turning on for a well-defined pulse and then going off again together. And it's just a single pulse. So this is what it looks like. You add the ethanol here, whoosh, goes on, whoosh, goes off. And then if you vary the amount of alcohol you use and you go down, you can see that you gradually vary the amplitude of this pulse. So this is a single synchronized pulse. So it shows the system doesn't really have to be noisy if it doesn't want to. But what's going on with this? So we call this pulse amplitude modulation as opposed to frequency modulation. And one thing that we thought about when we realized that the system was behaving this way was maybe what's going on is that since the cell reacts with a greater amplitude to larger steps in external stresses, it could be operating in a mode where what it's really responding to is actually the rate of change of the external stress. This turns out to be interesting. So in that view, if you did a step to a stress, you'd get a big response. But if you did a slow ramp to the same final level of stress, you might get a much lower response. Okay? we'd call that a rate-responsive system, a system that responds to the rate. And we wanted to set up experiments to test this idea. So we used a microfluidic system, and we set up either gradients, where we slowly ramp up the stress, or steps. And we compare the response of cells in those two different kinds of dynamic environments. So this is what it looks like if you do a relatively quick 50-minute ramp from 0 to 2% ethanol. You get pulses in both the gradient and the instant step. Okay? Now, if you make the ramp a little slower, you see that the response of the system goes down. If you make the ramp even slower, you see you get a much lower response, and even slower, you get essentially no response. So think about that. These cells, at the end, are in exactly the same level of ethanol. In one case, they've made a big response in sigma B. In the other case, they've made no response. And the only difference was how fast this stress appeared. Okay? And so here's a plot just showing how at fast ramps, you get a much stronger response. Okay. So what is this telling us about a general stress response pathway? What is a general stress response pathway for anyway? Okay, I think we always take it for granted that the cell should have some general stress response to respond to just general stuff. But if you really think about it, why does the cell even need that? Because in fact, there are more specific pathways that respond to particular stresses. For example, the environmental stress response in sigma B responds to both salt and ethanol. But there are pathways that can activate salt-specific genes in response to salt, and ethanol-specific genes in response to ethanol, even without sigma B. Okay? So what is sigma B doing? And how does it relate to this rate responsiveness? Well, here's the idea. If we do a slow ramp in the stress, there's no activation of sigma B. And instead, you specifically activate the specific stresses. If we do a fast step, then we still activate the specific pathways, but we also activate sigma B, which cross-activates everybody else, and many other stresses that are not drawn here. So the idea here, in a way, is that the general stress response is a way for the cell to freak out if things are getting bad rapidly, but to not do anything if things are getting bad more slowly. Okay? So it broadens the genetic diversity of a stress response only when conditions change rapidly. And one way to look at this is to actually do RNA-seq and analyze all the different target genes, or all the different genes in the cell in response to either a ramp or a step. That's what's shown here. And what you see is indeed the genes that are activated preferentially in response to fast steps in stress are in fact the sigma B targets. So that kind of underscores this point. Okay, so this is the idea then. At slow rates of increase in stress, you get independent specific responses, and at fast stresses, you can start to get broader and more overlapping responses in the cell. And I think an interesting question is whether this is what's going on in lots of other stress response pathways as well, where normally we don't really look at slow stresses. Most of the experiments we do, we hit them suddenly with the stress, we induce everything, and then we analyze. So I think it raises an interesting question. Okay. Well, the third and final thing I wanted to talk about today was a different functional role for pulses in the cell. And this is work that my grad student Joe Levine started in the lab. And you can see he's somebody who likes to move fast. But the actual project was really about how to go very, very slowly. And this starts out with, I think, a very basic thing that cells can do, one of the basic capabilities of cells, which is that they can react to a change in conditions after proliferating for multiple cell cycles. The classic example of this is in development, where you have oligodendrocyte precursors that were shown by Martin Raff and other people, that if you take them out of a developing embryo and put them in a dish, they'll divide for the same number of times in the dish before differentiating as they would have divided in the embryo itself, suggesting that they have a cell-autonomous timer internally that can keep track of time for a long, for many, many cell cycles, and then respond. Okay? So how do you make a timer? Now, the case we're going to look at is in bacillus, and what Joe discovered is if he just took these cells and he put them on a piece of agarose where they were nutrient limited, so conditions that generally induce this switch from being a vegetative cell into being a spore, and you can see the spores are these white objects here. Let me just play that again. If he did that, what he found was that the cell would divide about 5 times. So there'd be about 5 cell cycles, and then they would sporulate. Okay? And you can see that in this histogram here. So that's the distribution of cell cycle times. Okay? So it's not a stochastic process. It's not a Poisson process where they have an equal probability per unit time to do it. They're really waiting. And if you follow the bacterial literature, you know that a major theme in bacteria is the use of quorum sensing systems, where cells can build up concentrations of signaling molecules in the extracellular environment. And that would seem to be a good candidate. But through some very creative experiments that Joe came up with, he ruled out quorum sensing. And he also ruled out another mechanism, which is you might think that the cells are just gradually running out of gas. You suddenly put them on the nutrient-limited condition. They have some internal nutrient levels, and those slowly deplete over 5 cell cycles until they can't take it anymore, and then they sporulate. But it turns out you can rule that out, too. And you're left with the conclusion that they must be using some kind of internal cell-autonomous timer that's based on the accumulation of a regulatory molecule. Now, why do I find this a hard problem? Why isn't that no big deal to just wait 5 cell cycles? Here's why. Most genetic circuits that you would write down have the problem that they tend to equilibrate over a time scale of about 1 or 2 cell cycles. Why is that? That's because when cells are growing and dividing, the contents of the cell, all the proteins and everything else, turn over with an effective half-life of 1 cell cycle or faster. And so that cell cycle really sets a kind of limiting time scale. So how do cells defer differentiation for more than 5 cell cycles, given that they have this intrinsic time scale? Now, it turns out sporulation is controlled by a complex circuit that's been very well studied. And I'm showing here a tiny, tiny fraction of all of the components and interactions that people know about. It's highly simplified, but I just wanted to focus on the components of the circuit that we think are really important for this timer behavior. And the idea is that there is a set of kinases in the cell that respond to inputs from nutrient limitation and a variety of other signals. And when they're active, they phosphorylate a master regulator called SPO0A. And SPO0A, when it's phosphorylated, is necessary and sufficient to induce sporulation. Now, there's a second part of this circuit. The second part is that this SPO0A, the master regulator, can actually feed back and upregulate the kinase. That's this positive feedback loop here. Okay. Now, if you look at this again, you look at the activity of SPO0A in movies, what you see is striking, and we didn't expect, which is that there's pulses of phosphorylation and therefore pulses of activity of this master regulator SPO0A. These pulses are different than the pulses I showed you before in that they progressively build up in amplitude from 1 cell cycle to the next. You get about 1 pulse per cell cycle, and they kind of boop, 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 boop. They step up. And so only at the end do you get high enough SPO0A levels to induce sporulation. So this starts to look like something that might have a role in the timer. Okay. So we can say this is kind of a pulsed positive feedback loop. So that's the pulsing. And then we want to know, what's the role of the positive feedback? So one thing we noticed, as I just said, is that the pulses grow systematically. And it turns out that's dependent on the positive feedback. The way we know that is we can knock out the positive feedback, replace the kinase under IPTG controls at a constant level, and then we can ask, what happens? So if we plot the amplitude of one pulse versus the amplitude of the preceding pulse in the wild-type system, we see this systematic growth, which is why all those points are above the diagonal. If we do this where we knock out the feedback, we see the same distribution of pulse heights, but no systematic growth. So the feedback is necessary for this systematic building up of kinase activity. Now, you can say, well, what happens at the level of sporulation? And here again, the positive feedback is necessary for the timer. If you take this open loop strain where you've knocked out the feedback loop, and now you give it too much kinase, they sporulate immediately. If you give it too little kinase, they never sporulate. And somewhere in between, you get to a regime where they still don't have a timer, but they do sort of stochastically sporulate, okay, more like a Poisson distribution. So without this positive feedback, you can't have a timer. Okay. So these and other things are really what we learned about the system. And then we tried to understand, what is this all, how can this work? Okay. And here's the idea. We think the circuit is working in what we're calling a polyphasic feedback regime. The idea is that this feedback loop has 2 arms, and they're not active at the same time. They're alternating. During a pulse, you have phosphorylation. The kinase is actively phosphorylating the master regulator. But then that pulse is short, and only after that pulse ends does the kinase fully build up to its new level. And because the kinase is now higher, in the next cell cycle, the pulse, the phosphopulse, will be larger. And because of that larger phosphopulse, that will lead to an even larger step in kinase. So you get this kind of ratcheting up of kinase, which is in red here, and therefore phosphorylation activity from one cell cycle to the next. Now, what's good about this design? Why would you do this crazy design of breaking up a feedback loop in time this way? What's interesting about it is it gives you the benefit of positive feedback. The benefit of positive feedback is that a positive feedback loop can overcome the problem I told you about before of this limiting time scale due to cell cycles. Positive feedback can give you long time scales. But positive feedback by itself is not good enough because it's very, very sensitive. The time scale that a system responds on with just a continuous positive feedback loop, really, to get a long time scale, you have to tune the production and degradation rates of the component to be very, very close to each other. It's a very, very sensitive way to do it. It doesn't work well. But when you break up the positive feedback loop in time this way, you get a system that has the ability to access these long time scales because of the positive feedback, but with reduced sensitivity, so it can actually be controlled. That's shown here from a simulation where we compare the open loop system, or if we vary the strength of the positive feedback or the strength of the kinase expression level, we see that to tune it to a 5 cell cycle delay is very hard. You have to tune it very precisely right here. But if we do this pulse positive feedback loop, we get a much gentler tuning curve. So we think this is a basic principle that cells could be using to access very long timescales, but still be able to control events on those timescales. OK. So I think the main message that this work has, in a way, is that there are a lot of processes in cells that are fundamentally based on dynamic, often stochastic, and often unsynchronized behaviors between different cells. So this can be very worrisome if you analyze systems based only on cell population averages. So in the examples I showed today, we see that in the general stress response in bacteria, which is a really highly conserved, very basic system in cells, we have a DC to AC converter that converts things into stochastic pulses. We have rate responsiveness, which actually serves to, which we think is what the general stress response is really trying to do, which is to say broaden the response to very rapidly changing stresses. And finally, this idea of pulsing a positive feedback loop in order to control events on long time scales. Okay. And let me just thank the people whose projects these were. So this is James Locke, who just started he was a postdoc in the lab. He just started his own lab at the Sainsbury Institute, a new plant biology institute in Cambridge University. This is John Young, who's actually an MD PhD with UCLA and Caltech and just went back to the MD part. And this is Joe Levine, who did this timer. And grad student in the lab. And actually, our interest in all of these questions about pulsing really started with some work we did in 2008 in yeast, where Long Kai and Shiraj Dalal, shown here, really discovered this behavior of stochastic pulsing in the nuclear localization of transcription factors. So thanks very much. Okay.
Well, thank you for a fascinating talk. I'd like the next speaker to set up. And we have time for questions, so please approach the mic.
Sigma B? Yeah. Suppose 0A. Yeah. Yeah, so the question really is, what is the mechanism that generates and controls the pulsing of phosphorylation? And I have to say the answer is we don't know. We tried a whole list of different candidates, and we have a paragraph that Joe calls the parade of negative results, so we don't know. But we're very interested in that question. Yeah. Yes. So the question is, yeah. Yes. In other words, what we think the whole point of this is that we think that by making the cells larger, we're reducing fluctuations as the square root of n, exactly as you said, and that's the idea of using the square root of n effect to reduce noise without perturbing the cell in any specific way. Yeah. Yeah, so what we think is that there are fewer pulse events because you don't have as much fluctuation to initiate the pulses. Exactly. Yeah. Right. So good. So the question is a very good one, which is why do this? Why pulse? And first of all, whether it's specific to prokaryotes, I think the answer is clearly no, because we have p53, we have NF-kappa B, we have a whole range, we have crazy one, we have a whole range of systems that are doing this in eukaryotic as well as prokaryotic cells. Now, the other question is sort of what's the functional role of it? And I think there's going to be multiple roles. In yeast, we figured out one role, which is that by controlling the frequency of pulses, cells can control the fraction of time that all of the target genes of a transcription factor are activated. And by doing that, they can get a coordinated response where all target genes are maintained in fixed proportions, even if they have different characteristics of their target promoters. In other words, the cell doesn't need to fine-tune all the target promoters to have exactly the same response functions to get a coordinated response where all the target genes are expressed in fixed proportions. And I think from work we're doing now that I didn't have time to talk about, that there's a bunch of other functions for this as well. So.
I'm wondering whether there may be another explanation, which is that because of the homeostatic machinery of the cell, if you have non-pulse effect, it tends to go down to a glucose very quickly. And through a pulse effect, you actually can induce kinetics much more efficiently than through a ramp.
Yeah. So one question is if there's nonlinearities, those kinds of effects are possible, and we think they may play a role in other systems that we're looking at. Yeah. Good point.
Okay.
So the question is, do you need some kind of very complex network to make pulsing, or is it something that can happen just with a few proteins? So I think what this example shows is that the mechanism for generating pulsing does not have to be complex. You know, you can have a phospho switch and a feedback loop, and that's sufficient. Now, I guess the other part of your question is there just a correlation where you see more complex systems, let's say p53, more likely to pulse? And I think the difficulty in answering that question is that, for the most part, we have no idea what kind of pulse dynamics are going on, because this is a phenomena that can only be observed, really, using movies. And so I think as a community, we're just beginning to use movies to look at a wide variety of systems, and I think that's what we're going to find out, is how, how penetrant this kind of dynamics is across different systems. Good question. Yeah.
So looking at the short-term versus the long-term effects of stress response, I'm curious to get your thoughts on how we study antibiotics. So, usually we'll just throw an antibiotic into the culture and then look at the effects and make all these conclusions, right? So have you looked at different strategies of antibiotics and if they actually affect the way that the cells are killed and so on and so forth?
Yeah, so it's a good question. Antibiotics also constitute a class of perturbations that you can think of as stresses. And in fact, if you take Bacillus, there's some of these other alternative sigma factors, not sigma B, but other ones that do respond to different kinds of antibiotics. And so this is something now that we're actually doing. I don't have any answer, but we are looking now across all the other sigma factors to see what's going on. And I think that is a really good question, actually. Yeah.
Quick question. So what kind of predictive models you have for predicting the location of these pulses? Because, for example, can you assume they are like Poisson and you can relate the rate of these pulses to the input expression levels? Or what kind of models do you have for your framework? So
I understand, right, the question is, what is the model that relates input signals to pulsatile behavior in different systems? Yeah. So, in yeast, there's systems where inputs affect frequency of pulsing. In sigma B, input affects frequency of pulsing. But I think there's going to be other systems where it might affect the durations of pulses, the amplitudes of pulses. I mean, you see here in sporulation, the system itself is building up amplitude. So I think the thing with this kind of dynamic signal encoding is that the system can encode signals in different characteristics of the pulsing. And so I think one of the questions is, are cells using that to effectively expand the bandwidth of a signaling pathway and multiplex signals onto a single molecular component, if that answers your question. Is that? Yeah, good question.
So thank you. Please